Welcome to this first plenary lecture today, and I have the pleasure to introduce you Stéphane Avril. Stéphane Avril is a distinguished full professor at the Institut Mainz Telecom, affiliated at Mainz and Etienne in France, and he runs a group in soft tissue biomechanics with a special focus on constitutive modeling and identification using imaging techniques. He was the director of the CIS Center for Biomedical and Healthcare Engineering between 2010 and 2020, and has been the deputy director of Symbios since 2016. Regarding his background, Stéphane received his PhD in mechanical and civil engineering in 2002 at Mainz and Etienne. And after positions at Arts Métiers Paris Tech and Louvain University in UK, Stéphane returned to France in 2008 and extended his broad experience of inverse problems to soft tissue biomechanics, especially regarding aortic aneurysms, in close collaboration with vascular surgeons. Also, Stéphane was visiting professor at University of Michigan in 2008 and has been visiting professor at Yale University since 2014 and has received many awards over these years and distinctions, led 2 national and air grants in soft tissue biomechanics and supervised more than 25 PhD students, quite a lot. And in 2050, Stéphane was awarded with an ERC consideration grant to study the localization in biomechanics and mechanobiology of aneurysms. As you can see, most of the Stéphane's research is aimed at improving the treatment of cardiovascular diseases by assisting physicians and surgeons with biomechanical numerical simulations. Actually, in 2017, Stéphane co-founded PrediSurge, a spin-off company of IMT at Mainz and Etienne, that offers innovative software solutions for patient-specific numerical simulation of surgical procedures. And in fact, today, Stéphane is going to talk about multiscale modeling, tissue characterization, and inverse methods toward the Aortic Digital Twin. So I think that it's, maybe I can stop talking, and Stéphane, is all yours.
Thank you, Jordi, for a kind introduction. So I think yeah, it was a lot of words, and nowadays actually I'm working like between France and also Austria, where I am at the moment at TU Wien, and most of the talk is going more to be related to research we've done so far on aortic aneurysms. So this is supposed to be a lecture, so I hope I will not be too long, but I try maybe to be to take some time to explain things, and so it can even be stopped by anyone who would like to ask a question during the talk. So I would like to answer some of the questions that are written here, like, can continuum mechanics model predict human health? And then go deeper and deeper in what we've done on that field related to aortic aneurysms, especially aneurysms of the ascending thoracic aorta. So, of course, the first question about continuum mechanics model. So I'm more a solid mechanics guy, and Irene was presenting before is more from the fluids side. I think that we still have, I mean, a lot of efforts to do to combine, I mean, efforts from both sides, because usually students are taught in different master degrees, fluids and solids, but still, yeah, there are some connections. So but it's all continuum mechanics, and I wanted just to mention that nowadays continuum mechanics helps to take decisions every day. You all know these companies, so if you don't know mine, PrediSurge, I'm going to give some words about that, but you know also the others, The most successful one is, of course, the one heart flow. Of course, it's not only based on continuum mechanics, it's also using a lot of artificial intelligence, but it's kind of I mean, already big success for mechanics. So my own experience, as I said, is is related to aortic aneurysms, and it has always been the result of strong collaborations with clinicians, always sitting together, discussing. I think that was well explained also by Irene, that I mean, the combination of our worlds needs hours of talking, and that's why I show this picture here. And the picture led actually to the foundation of PrediSurge. I'd like to give some word about it. So first, in case some of you are not familiar with aortic aneurysms, so as I said, I will focus mostly on aneurysms of the ascending thoracic aorta, but I'm going also to talk about aneurysms of other parts of the aorta. All these aneurysms are devastated they can lead to devastating complications, because you can live with an aneurysm, but the problem is that the weakening that it brings to the wall of the artery is prone to cause a rupture, and then a rupture for the main aorta, for the main artery of our body, is almost always fatal. Okay, so well, what we did for I mean, to get to something which is concrete and has applications every day for surgeons, is to provide planification, sizing of stent grafts, which are used to treat these aneurysms. So the surgeons, they bring these stent grafts from catheter, so it's mini invasive surgery in a way. This is called EVAR. But the problem is that when the arteries, the aneurysm of the aorta covers some branches like the renal arteries, then the stent grafts, they need to be customized with holes that we call fenestrations. And so PrediSurge made a software based on reading images and processing them and solving a finite element model of the stent graft deployed in the artery in order to find where the holes should be, and then doing that automatically to provide like a map of the device that can be manufactured, and saving like this several weeks that the patient has to solve. So I wanted to mention that because this is something that has occupied us for a long time. We started with an ANR project with other partners in France, and we simulated interactions between devices like guide wires and also stent grafts in the aorta, taking into account the properties of the aorta. But the main thing was that we went up to validation through a lot of patients, clinical cohorts. So the results have been published. And so the comparison was made, for instance, here with this collaboration with Marie Lanelong in Paris, where the stent grafts that had been proposed by experienced surgeons were compared to the ones that were predicted with our software, and showing that there was a very good match, and all the very few complications that occurred during the surgery based on the surgeon predictions, they were all different with our simulations, which doesn't show that they are better, but just shows that well, there is probably some new validation study to be carried out in order to also improve not only the time that was the main criterion, saving time for the surgeons to get the stent-graft, but also gaining precision in. So we were able to show that we are as precise, but saving precision, gaining precision is even probably something that we can prove, but it's very difficult to set up a clinical study where we can really show it. Okay, that's that was my kind of introduction about, okay, so what's the state of the art, things that we really find now in the industry. I'm a researcher, so I mean, my research is in a lab in the university, and so what we are doing now is more about what could be in future products in these companies. And so we started to work on deriving mechanical factors in order to predict aneurysm risk rupture, aneurysm rupture risk, okay? So especially for that part of the aneurysm, because that part we cannot, you have to know that that part we cannot use EVAR. There's no EVAR already on the market that can treat this these aneurysms. So this has to be open. And open surgery is still very heavy surgery that has a lot of, that can even bring some complications afterwards, like 10% of a patient after they have difficulties recovering. So it means that taking the decision to treat an aneurysm in the ascending thoracic aorta needs to be decided with a lot of care, and biomechanics can help, because some aneurysms that are have been found to have a very low risk of rupture, even if they are big. Okay, and so now just the size is taken into account by the surgeons to take the decision. So we did a lot of mechanical tests, actually, receiving aneurysms from the hospital, and then they look like this, then opening and cutting squares, and then inflating them. So we started, we were the first to do these tests in order to have a rupture of a bit of aneurysm, to have a rupture in a biaxial loading, because a lot of tests have been done in the literature using uniaxial loading, which is not, I mean, providing these conditions that you find in vivo. And so we also did something that was very interesting, and that relates to all the machine learning that we're trying now to implement in all the fields of biomechanics. You need, as Jérôme said before, a lot of data. So as we cannot have a lot of specimens, we decided that with a single specimen, we should have a lot of data. So instead of measuring just like the stress in the middle, or instead of measuring the strain in the middle, we painted dots on our samples. And then like this, we could track the strain everywhere and get like thousands of data with a single sample. So how did we do that? Actually, thanks to digital image correlation and these cameras here, we could track the strain of every point and also their position during the inflation. And like this, we were able to reconstruct the surface of the sample during the inflation. So that's the work that was done by this postdoc student with the Whitaker Foundation funding. And then what was very interesting is that we also use some isostatic principle in order to reconstruct the stress everywhere, so that at every point, for thousands of points on a sample, we could plot a stress strain curve and define a point where the sample would have been when it was in vivo by extrapolating the strain towards the one that we got from the CT scan of a patient, and also knowing what is the maximum stress and the maximum strain that this tissue at that precise location could have taken. So with that, we were able to train a machine learning model in order to find the location of rupture. So how did we do that? Actually, we took like the thousands of curves on 20 samples that we got, and then using all these data, we knew which were the points where the rupture had occurred. So like here you see that sample, the rupture was here. This is a picture just at the end when the rupture occurred, and the same here where the rupture was here. It was here and here. And then we trained, and then after we evaluated for other samples that were not used for the training, if the prediction of a rupture was correct with a risk factor criterion, that when it reaches 1, it's the rupture, and it was always correct. And that's kind of interesting because a lot of predictions were based so far on the stress, local stress, but we found that the stress can miss, for instance, on that sample, the maximum stress can miss the location of rupture. Okay, so which makes sense mechanically. This is not only machine learning black box. This is based on the stress strain curve, and actually it relates the rupture where it occurs to the stiffness during the test, and then the stiffness relates to how much we straight the collagen fibers inside the tissue. And if you straight them earlier, then you're going to reach the rupture earlier. And so that's what we found, even if then after the machine learning was able to do it by itself. But we found it, and then this correlated very well with basic principles that have been known by surgeons for a long time. Like for instance, here in this paper, we refer to, I mean, something that is told by surgeons to their patients having an aneurysm. So don't do what you see on this picture. Don't do weightlifting. Don't do anything that may create some big variation of your blood volumes in your aorta, because then locally, if your aorta is stiffer, then it will not be able to take this change of blood volume. And this is what will cause the rupture, okay? Because locally, if your aorta is stiffer, then it will straight these collagen fibers and cause the rupture earlier. Okay, so this is what the machine learning model predicts. I mean, on an automatic way, but that we found also from mechanics principles. So that was the early work that we did doing this test. But then we wanted to go further and to couple mechanics with biology. And so that's like the transition between biomechanics to mechanobiology, where biomechanics, you take like I mean, we tested these aneurysms as we would have tested rubber, and then or any industrial material, and then apply the stress strain or stress analysis principles. But aneurysm progresses, and the problem is more to know when it's going to progress too far and create some real risk for the patient. So that comes to be quite some more difficult problems, actually, but they are very important because they need to they need to be known for decision making for the surgeons. So I think that for that we did some. I think I liked also in Irene's presentation the question about the what if questions, and also taking some causes, and then I mean employing some cause consequence principles and try to simulate what they give, not claiming that we would be able to evaluate the progression for all the aneurysms. That's something that is more for the future. I'll give some clues about that later. But so one thing that we started with was that it was shown that there is a correlation between hemodynamics and these aortic ascending thoracic aortic aneurysms. So a lot of the patients developing these aneurysms, they have a disturbed hemodynamics. They have a jet flow. You have to know that half of the patients having these aneurysms, they have a bicuspid valve, which is, although very rare, it's only for like 1% of people, but a lot of them, they develop these aneurysms, and a lot of them, this is related to the disturbed hemodynamics. And it was shown by some researchers that the location where the jet flow impinges on the wall for these patients is a location of elevated wall shear stress, but also of damaged extracellular matrix in the wall of the aorta. So there are some biological pathways triggered by the effects related to the hemodynamics that leads to an overexpression of matrix metalloproteases at that location that leads to what we call an injury, a proteolytic injury that is a kind of effect, a cause that may have some effect. So that's something that we can model by using some principles that I'm going to explain here. So if you have a stress strain curve of a tissue that we take from, from an aorta, then it would look like this, okay? But then if you have this injury, then it would break some of the elastin fibers, or it would damage the wall, and then it would lead to that red curve. So the only thing is that we do not have here, and that we wanted to model, is that there will be an adaptation of the cells in the wall to that new situation. And then this is what is called healing, that the cells will try to repair the tissue, but repairing with the new situation will lead to a new geometry that is that leads to this change of diameter. And that's something that we started to model using the models developed by Jay Humphrey, and I think they were mentioned also by Irene earlier. So these models are based on similar equations as the one that we use for rubber and for all the materials that can be extended or distended on large strains. So based on the theory of hyperelasticity that we call in mechanics. But the thing is that it is modeled as a mixture, so like a composite material. And actually, we use like, we model the artery like a composite material with a matrix. So the matrix is modeled with this equation that is a Neuhoeken strain energy density, which is like the simplest isotropic model for a rubber, for instance. And then we have these fibers that model all the network of fibers that we have in the arterial tissues, including collagen fibers, but not only, and also their crosslinks. And they are based on exponential functions that they were found first by Fung, and then after, like Holzapfel developed the model. So it's based on the similar theory. And so in the constraint mixture model, you have this word constraint. So this means that we take into account the fact that in any reference configuration, you will never find any of these constituents. So neither the matrix that here contains most of the elastin, nor the collagen or the smooth muscle cell, you will never find them with a similar stretch, because it's like the elastin has been deformed during the growth of our body because it was created early in life. The collagen is changed all the time. And so this creates what we call residual stresses in a tissue when you remove it from the body, because all these constituents, they do not have strains that are compatible if they are stress-free. So here I designed them like stress-free, for instance, and then we have to take into account this pre-stretch to all of them to put them in the model of the aorta, and then to after consider the loads. So that's the model that was developed quite a long time ago. And this model can consider that some constituents, like for instance, the fibers, the collagen fibers, they can be removed and also created. And then we have to consider the turnover of them. And for that, we need to change first their mass and also their pretension. So the mass is usually based on a principle that you have in the tissue, the need of reaching some homeostatic stress. So this means that if a tissue has a stress that deviates from the homeostatic stress, then it will change the mass of the collagen fibers in order to restore this homeostatic stress. But there may be other reasons why there would be creation or a removal of fibers, like, as I said, like the proteolytic injury, but it can be also an inflammation. So then the model just takes into account that we can change actually here the mass of a different constituent, and then this will affect the total strain energy density of the tissue. And there is also remodeling, that actually if the collagen fibers are changed, but that the tissue had been loaded in before, then the new pre-stretch of the collagen is going to be different as the one as it was like before an aneurysm occurred. So it's like the collagen, they always want to maintain the same stretch. So if you dilate the aorta, then the collagen, they will work to, remove the fibers that have the very large stretch and put new fibers that have the homeostatic stretch. And then this creates this kind of, kind of creep that occurs actually, and that is what we observe in this aneurysm progression for patients having these aneurysms. So that's what we modeled, and I just explained before the constitutive equations, but then after this has to be implemented in a finite element framework. So all the models, as I said, were developed quite a long time ago, and they were proposed by J. Humphrey, but we struggled a lot to implement them in a finite element framework for patient-specific aortas, because most of the time they had been used for cylinders, and but patient-specific aorta brought some issues related to numerical stability, because these models have, one of the main issue of these models is this pre-stretch that you have to apply everywhere, and that can create some instability issues that we had to overcome by playing around with different initial pre-stretch until finding like a good combination of them for the different constituents. So that's something that is explained in our papers here, but that was the main innovation of our work in that field, because, as I said, the models were quite old before. And so then we applied, the cause that I explained at the beginning, that we have clearly patients having this jet flow impinging in a specific region. So we can model that there is a loss of elastin at a given time. So localized in time and localized in space here. And then let the model, using the principle I explained before, predict what's going to happen. And this is here what happens like during 10 years after inducing what we call this insult. And you see that the very interesting things, so this is for patient-specific shape that was reconstructed from CT scan, that the first thing is that the aneurysm grows everywhere. And that was the first lesson we learned, that even if you have like a localized degradation of the matrix here, then the reaction is a complete change of geometry, just because this principle of homeostatic stress will be affected everywhere, because if you change the stress here, then you change the whole stress distribution all around. So that was the first thing. But even if we found that, for instance, most of the collagen change of density was localized here, also a change of thickness. So that were the predictions, but these predictions should not be the same for all the patients. So one thing that we started to think about is how can we take into account with these models that every patient is different? So that's something that we're still working on. But the first thing I wanted to show today is that we could take into account some genetic factors that I will introduce related to smooth muscle cells in the wall of the aorta. So I'm going to explain some of the studies we do at that scale later. But here, I take that into account at the tissue level. So at the tissue level, we may have patients having smooth muscle cells that do not mechanotransduce the stress similarly as in other patients. And these principles that I explained at the beginning, that the change of collagen density would be related to the deviation from a homeostatic stress in the tissue, then we can introduce a factor that would tell how much the cell missense the stress. And if this factor is 1, there is no missense of stress. And you see that actually for the patient a patient would have, this factor, aneurysm would grow, but the stiffness of the tissue would not change, okay? Because the collagen fibers would always try to work at keeping the same tension. But if we have this missense that exists, and this is what occurs in patients, I'll explain, because some of the smooth muscle cells, they change their phenotype and they turn into synthetic phenotype to make new collagen fibers, and then they do not really work at sensing the stress, if sensing ever means something, but that's something we can discuss. Then we see that in that case, we would have an increase of stiffness, just because actually when the smooth muscle cells would take larger tensions. So that's really an interesting effect that we could, for instance, define subgroups of patients and include information on that in order to predict different types of reactions. But then we get into, I think, something that started to be discussed today, and that is probably one of the main limitations of the model so far that I presented, is that it can, if you have all the assumptions about the patient and you know everything, then you can predict what's going to happen. But we will obviously miss something about the patient, like boundary conditions that we do not know, that we don't know this genetic factor, or how could we know? So I think that the future of these models and the future of many of the models that we develop in our field, they need to be interfaced with data-driven modeling. That's obvious, and well, I just want to illustrate that for our case. So I just cite a paper I really like. I think that this was written by after an EU project where related to the heart, and I admire this work because it really shows how to combine mechanistic models as the one that I introduced before, and a statistical model that can well, still improve the applicability of the mechanistic models that we have here, because to work on a real patient, it's better, and to provide some information to a clinician is better to give some probability information than to give a kind of deterministic number that doesn't mean anything. So we started to work on that by developing a kind of framework where we, for all the patients that have an aneurysm at the hospital, before having their surgery, we have them taking a 4D flow MRI so that we can derive for them the blood flows. And then at the same time, we take the tissue and test the tissue in the lab. And so start to build this database that we can work with so that we have a large number of informations about the blood flow and the tissue properties, and well, this is still a work in progress. So this is just here I show the workflow, but I mean the results of that will still take a number of years, I would say, in order to have a database big enough. And that's also something I'd like to do, is to kind of also involve other groups. Like in France, the group of Dijon, for instance, is also doing the same, and try to involve even more so that we can speed up I mean the creation of that database. Okay, and yeah, so this is also something that we try to include in the database. This is tracking the motion of the aorta during the cardiac cycle in order to derive stiffness properties, because as I said before, the stiffness is for us a critical parameter, but this is still something that has not been derived in a lot of patients, and so it's important that we could also have it in the database. And yeah, and the boundary conditions are probably also very important, but this is still something that our models kind of have difficulties to really take into account. So when I say boundary conditions, it's just that even in our fluid structure interactions model of the ascending thoracic aorta, so we have difficulties in taking into account the motion of all the surrounding, it's of the heart and the arteries around, and so that's also something that well, I'd be happy to discuss about some clues that people would have on that topic. But when you see the complexity of the rupture that occurs during a dissection, I think that this is also critical. So I'd like to move on to another part of this study that actually I think that what I said is that, okay, so most of the problem that we have that we cannot solve with mechanics, maybe more related to biology, we would maybe expect that machine learning could give us some clues to solve them, but still we decided that also we should try to do some continuum mechanics at the cellular and subcellular level, and try to get also some basic principles of continuum mechanics that could also help us in evolving our models. So, well, I think that we did that also thanks to the influence of Jay Humphrey, who has been really a mentor for us on that, and while he cites very often that this citation that dates back from the antiquity, and that well, tells that you cannot understand a tissue if you don't know from where it comes from. Okay, so so developmental biology is very important, and so we try to work more and more with biologists and to understand so why the cells that we have in the tissue, they would behave in a way or another. So how they keep history. I mean. How they are influenced by their history in order to be able to take that into account in our models. So this is very important because like there are many effects in aneurysms that take place first at the cellular level, and this has been shown again by Jay Humphrey, where they found that actually about 25 genetic mutations that affect smooth muscle cells and their missensing, they always lead to aneurysms. So that has been found. So patients having these 25 genetic mutations, they always have an aneurysm of the ascending thoracic aorta. And all these genetic mutations, they have like an animal model, a mice model that has been developed, and the mice, they develop the aneurysm. So this is really important to understand why just a gene change, and sometimes not even another big change in another tissue, but there is an aneurysm. So this is this is important to understand that. So that's why we started to work on understanding the mechanics of the smooth muscle cells. And so as we had the tissues in the lab coming from the hospital, so we decided to take the smooth muscle cells from the tissues and to investigate first the tension that they apply on the tissues, and where we cannot mimic the tissues, so we use gels. So we cultured the cells coming from aneurysm patients on hydrogels, and then using digital image correlation, we measured the forces that they apply. So it is a famous technique of traction force microscopy, and we did that on healthy, also for healthy patients. So I was not able to take my cells, as you may understand, but there is a data bank in Paris where also aortas and heart of healthy patients were collected for dead people just after death, still fresh. And so we were able to compare. And one thing I'd like to show is it is really the result that what we found is that aneurysm, they apply larger forces, significantly enough to be noted, than healthy cells, healthy smooth muscle cells. So this is something that actually we were able to relate to the size, the average size of the cells in aneurysms. We found that there is a phenotype that occurs only in aneurysms, or that is kind of developing more in aneurysm, of stronger and bigger smooth muscle cells that we believe is a kind of change of phenotype towards more like a myofibroblast phenotype. And this is very interesting because we do not see that much change in the cytoskeleton of the cells when we on so much difference when we do AFM indentation. But still, if we could find a way of having some clue on the existence of this phenotype in the patient, that would be very interesting, because this relates to I mean, something that occurs more in aneurysms, and also that can be related to the tissue properties, because actually, well, there are some, I mean, a lot of mechanobiological studies, and actually in Barcelona, there is a very strong group also doing mechanobiology on the cytoskeleton that relates the tension in the cytoskeleton of the cell that we measured to the tissue properties that or the matrix properties that is around. And that's really something that we'd like to investigate more in the future, because, well, that was in the title of my presentation, and that's going to be almost the end now. I said we want to go towards a digital twin, okay? So the digital twin is a computational model, personalized, patient-specific, but not only like making predictions, but also taking information from the patient. Okay, so what kind of information can we take from a patient? So we got the information that the patient can go every year and take an MRI, right? But we need more information that can relate to biology, and so that's why we need to find ways of having information on the sensing, force sensing mechanobiology of the smooth muscle cells, because we know now that they are very important. And if we have information on them, then our model can predict the progression of the aneurysm, and they could also learn from also like the database that we are building from the patients. But yeah, so this is this loop that we need to create. So fortunately, we got last year Etienne project, and the objective is to build a digital twin of the aorta. So there are a lot of PhD students working on that. But yeah, it's probably take more than the time of a PhD to do that. And I'd like to finish with this very nice paper, which I think is going to be seminal. So by a consortium in the United States mostly, but there was also a team in France who participated, and they were able to combine a model like the one that I showed for growth and remodeling predictions with a database of data that were acquired on the effects of drugs on a different number of animal models. Okay, and so they were able to combine that in order to predict what would be the effect of these drugs on aneurysm. So other experiments that were not done before, but that the model predicted. And so now they are verifying that, for instance, that they found a drug that could affect the sensing of the smooth muscle cells, and that could arrest or slow down the growth of aortic aneurysms. So I think that, well, this is probably, again, the beginning of something, but every patient would be different. So it's still, I mean, something that, well, will require this complete model and taking information from the patients. But, well, this is the way to go. This is probably not that fast enough for the patient. This is also probably quite not that advanced compared to other fields that I know. For instance, Jérôme is working on intervertebral disc, and others here are working on the heart, and probably things that are well more advanced than what we do on the aneurysms. But, well, I think that it's nice also to have these forums like today at this summer school and have discussions and share our experience of different fields, working all for the benefit of the patients. So I thank you all for the attention, and thank everyone who contributed to the work I showed today. I'm happy to take questions now. Thank you.
So thank you very much, Stéphane, for this nice work. I don't know if anyone has any questions. Otherwise, I have one, but I don't know if any of my colleagues have questions. Okay, I see. Yeah, Jérôme. Yeah, Jérôme has a question. So,
yes, but please start, Jordi.
Sorry?
You can start, Jordi.
Yeah, well, there is Benjamin also that has questions, but okay, yeah, I can start. So my question was related on the FCI with aorta, and well, it's not a question. I would like to know your opinion, because at the end I have experience with the left atrium, not with the aorta, and my experience with the FCI in the left atrium is that at the end all the parameters that you need to set up this configuration, this biomechanics of the left atrium, it's quite difficult. There is not much research, or it's difficult to find those values in the literature, and then it changes a lot the results among the patients. So for me, it's like difficult to establish like a workflow that, okay, I'm going to apply these parameters like for all my cohort. So my question is, how is in aorta, if it's like very patient specific that you need to tune like every parameter for each case, or you see that in a close future you can be like more straightforward and have like a configuration for all the FCI simulations in aorta?
Okay, so you mean FSI, free structure interactions, or yes. FCI? Okay, so I think that in the aorta, so I have a person also in my group starting to work on the left atrium, and we work on that project with the same cardiologist, MR cardiologist, so who provides me this kind of images. So the difference, of course, is that in the left atrium you have a couple of voxels, and in the aorta you have many voxels. Okay, so in the aorta, from the 4D flow MRI, we have very good data nowadays, and so based on this data we can reconstruct our models, and we can assign boundary conditions that are patient-specific, informed by this data, and without too much effort. I think, well, one postdoc developed the framework, but then it's working quite well. What I see in the left atrium is that actually you have to augment the data a lot, because the data, the voxels are, I mean, the size of the voxel and the size of the atrium, I mean, the size of the atrium is of the left atrium is 10 times smaller maybe than what we have here. So that's, I think that's the main issue for the left atrium. But for us, as soon as we got this 4D flow MRI for a patient, then we have already very good data that we do augment them by using them as boundary conditions only, and then simulate the flow with CFD. So we don't use, for us, the next, the thing that we should do is be able to have more FSI with especially the motion that can be related to by coupling with at least the left ventricle. That's something that would probably give us more information, but that has still never been done so far, like coupling left ventricle and on the aorta, because you have like the aortic valve in the middle, and this is quite challenging. For us, that's probably something that would bring important knowledge. But for the left atrium, I think that augmenting the data because of not enough voxels is a challenge, as I understand. So it's probably your field, so you have maybe done things interesting.
Yeah, well, the thing, well, my doubt was more focused, I mean, because I don't know if from MRI it's enough to extract like all the mechanical properties that you need for an FSI.
Yeah, for the wall. You mean for the wall, for instance?
Yeah.
Yeah. So that's also something I understand. Yeah, because you cannot track easily the wall motion in the left atrium. So in our case, so for the wall properties, so the MRI is not good enough either. So for the flow MRI is not good to track the wall motion, and so we used CT scan gated multi-phase, so that we could this patient, they have it, so usually we can use it, but then it requires some multimodality. But what we found is that even with a CFD, we were able to have quite good agreement between the flow that we predict. So if we need only to predict the flow in the aorta, well, the wall properties is not that an issue for us. Okay. If we want to predict the motion of the wall and the stress in the wall, then we need, in that case, the wall properties. And so for that, yeah, combination with a CT scan is needed.
Thank you very much. Jerome, you had a question?
Well, I think we can give the words to Benjamin, who and then there's one question from City Kanta in the chat.
Okay, Benjamin, if you want.
Thank you very much, Stéphane, for the very interesting talk. I wanted to ask about, so for your GNR model, and determining material parameters for, the elastic material parameters is easy enough. You have your test data and you can fit your material model to that. But for the actual material parameters that pertain to growth, one would need some sort of data that model, well, data of how the material grows over time to fit a model. So, which I imagine would be difficult to obtain. So how do you deal with getting relevant material parameters for the growth part of the model?
Okay, yeah. So what we did now is which is the beginning, is that we found some growth rates that have been related to some morphological parameters or to body weight ratio, sex, age, this kind of thing that we found in the literature. That some people, they have like look at the evolution of aneurysm in many patients. So then we calibrated our models of the growth and remodeling model in order that we get the same that we get the growth rate of progression of the aneurysm similar to what has been found in this database. Okay, but this is still not patient specific. So then for the patient specific, what we plan to do is that we can have like a follow-up. When we have follow-up on a patient, then we can take from this patient the growth rates that we have from the previous year as a parameter, and then predict the next year with that growth rate. That would be more patient specific, but still not completely patient specific, because you never know if some new effect would occur. But yeah, that's how we plan to proceed in the future. And the other thing is, can we anticipate some change of rates? And so that for that, well, that's where we want to link hemodynamics and also stiffness to the growth rate, but this is still also a work in progress. So for now, okay, so the parameters that we have in the model are based on like the progression rates that we can have for similar group of patients.
Makes sense. Thank you.
Thank you. I saw that there was a question also about how to get the material properties from imaging. So this is something that I explained a little bit using the gated multi-phase CT scan. So where we have like motion of the wall that can be. So what we do is that we do mesh morphing. So we have a mesh for a phase of a CT scan, and then after we morph that mesh to the other phases, and then from the deformation then that we have to apply to make the morphing, we did use the strain, the local strain, and then we relate the local strain to the local stress that we assume to be related to the pressure with the Laplace law, and then after we get material properties like this for the wall of the aorta. That's I mean, in patient-specific way, and this requires the dynamic gated CT scan, because we tried to do it with 4D flow MRI, but the resolution was not good enough, and so well, we could only reconstruct like an average geometry, but not like geometry at every phase during the cardiac cycle.
Okay, so I don't know if there are more questions. Otherwise, I pass my word to Jerome.
Thank you, Jordi, and thank you, Stéphane, for this great talk. Stéphane, I have a question. First of all, I really liked the reasoning you did on the last slide, and I think that people, modelers in general, and so we need to be careful to not overpromise. That's an error that has been done in the past. And but also we need to prove that things work. So yeah, there is a trade-off of pushing towards the use of patient data and starting then to give recommendations, and also cautiousness, and be very very clear about the limitations, and very transparent. So so I really like then this this kind of reasoning that might seem obvious, but I think it's not too efficiently told in the community. My question is about this the definition of the homeostatic stress. Actually, homeostasis is not an intrinsic property, and it's a result of an emergent process in biology, and you might have different homeostatic conditions depending on the time of observation, depending on the biochemical environment of the cell, depending on the patient, etc. And I guess this is something that you have started to see when you do introduce then this correction function key, and so you have applied then this correction function key so to different patients. But then I'm wondering whether you have also tried to apply it locally, because of course you have gradients of conditions of everything in your in your tissue. So what do you think about this?
Yeah, so I think that you're right that we reach some critical point where I think that these very basic principles of biomechanics that date back, I mean, this homeostasis. So for the bone, there is this is principle of mechanostats, and I mean, this is the foundation of biomechanics, right? Okay, but then when we start to apply in computational model, and then to assign them to every Gauss point of a finite element model, then is it correct? That's your question. Is it correct to apply that? So of course, globally it works, but it shows that it still predicts an aneurysm growth that from an insult that is caused by something that really occurs in the patient. But then the local effects are probably kind of averaged in, and we should not over-interpret the local effects that we would observe from our model. So we need to use this model to predict something that is clinically meaningful. So maybe predict like what's going to be the diameter of the aneurysm in 5 years. Okay, well, maybe well, and you don't need you don't need a finite element model to do that, because if you take all the patients and then you take a machine learning model, you will you will get the same. But still, I think that you have some good explanation of a patient would grow very fast, okay, and machine learning would predict it as well if you have but with this model you would be able to understand why this patient would grow very fast. So maybe because this patient, we see that actually the synthesis of collagen in this patient, there is a problem or something like this. So I think that well, we can still predict something global with principles that are very fundamental, and that can tell something to the clinician. So I think that gathering all these conditions is important. So explaining and these models, they explain. So they are not probably true, as you say, locally. They are heavy globally, so that's why I think that machine learning can probably in a couple of years, I mean, make this model become old-fashioned, but not that not completely, because I think that they explain things, and we would still like, even when machine learning is able to make all the predictions from the database, we still might like one day to understand what's going on in this patient, okay? And then these models, I think that they can reveal, okay, so this is because in that patient, it would grow fast because, okay, the model says if an aneurysm grows fast, is because the collagen is not produced fast enough, for instance. That's, well, that's my understanding. That's why I'm still pushing in that direction of using these models. I think that they are based on principles that have been known for more than 100 years. In Wolf and Frost. I mean, this was for the bone, but this applies also for soft tissues.
Yeah, that's true. Thanks. And bit related then to that, to this no reasoning about homeostatic, whether global, whether not. I have a question regarding then the differences you've seen in the smooth muscle cells. Where the cells for patients with aneurysm, where the cells harvested at the site of the aneurysm or away from the site of aneurysm? Yeah.
So they were always harvested at the same location of the aneurysm. So that part, okay, so that part here, and so same location, and the same for patients without aneurysm. So same location. Actually, this the existence of the specific phenotype of phenotype that are overexpressed in aneurysms, biologists have already shown that, but the phenotype was more related to transcriptomics and how this manifests in terms of mechanics was not really done before, and so now our job is also to couple. So we are now working with phenotyping these stronger cells in terms of transcriptomics, and in order to find if there are some genes which are more expressed, and then that's also well, a new field also for us. I didn't do that before in my lab, so that's also I mean a difficulty. But I think that we should all, and I know you do that in your project. That's very important to couple transcriptomics because. With transcriptomics, because this should be in the digital twin, right? Thank you for your nice questions.
Thanks, thank you, Stéphane. Jordi,
I think that Liam has a question, but I don't know if we have time.
We have 3 minutes, and we will have eaten all the break. But we're here to discuss.
Cool. Hi. Thanks for the talk. Really fascinating. I'm interested in the bulge testing and the experimental method, and your opinion on how does, excising a piece of the vessel wall and then putting it into a disc for the bulge testing, does that significantly change the or affect or bring in uncertainty about the actual material property when you test it, given that it's now removed from the body and it's not in its natural configuration as a whole, or are those material properties and the stress strain curves quite reliable to then sort of extrapolate to the normal configuration?
So that's a good question. So what we're still concerned about is more how to use, but it comes to the same point, how to use the values that we obtain from these tests, because if you compare to if you use it from a stress analysis that you get for a real aneurysm, then this is not like the conditions for us that would really change the tissue behavior, but this is more how do we reach a rupture? What is the type of rupture that really occurs in vivo, actually? And well, you've seen this picture. I think that maybe you're familiar with. Okay, this is a CT scan of a dissection in the ascending thoracic aorta. Okay, so we did our test here. This is where we have the maximum tissue. So we rupture them. But actually, the dissection, it seems to be like more. I mean, first a delamination and then a rupture and also that goes more in a kind of helical way, or that propagates in kind of helical way. So, well, our concern is more that are we really? I mean, is the rupture of the aortic tissue really meaningful to predict anything that occurs in clinically? That's really the question. I think that well, our tests, they do not predict when this would occur. Okay, and this is what is observed in the clinical practice. Because if you run a stress analysis and you check when the stress here is going to reach the stress that we measured in our test, I don't know if that would tell something. So that's I think that's there's still a lot to do on more understanding why an aortic dissection starts. So we know that it starts because of delamination issues. Well, the link between the different layers of the artery is becoming more and more brittle. But well, so what is the critical strength for that? For me, this is still an open question.
Okay. And do you think the stress analysis from imaging might be able to elucidate more information with regards to, you could almost have real-time measuring of the stress while prior to it about to get dissect or later dissect or rupturing? So
I think that what is important is that there may be still what I think we can learn from what we did is that there is so maybe what matters a lot is the strength, and of course the rupture is always this ratio between stress and strength. Okay, but the stress, except is you have these gigantic aneurysms, but usually the stress is not like, I mean, changing a lot from one patient to another. It's increasing when the aneurysm grows, but what changes a lot from one patient to another is the strength. And so the strength is related to how the tissue properties are good or not in a way. And so what I would believe is that if you have a tissue that has, I mean, tensile properties or properties in biaxial tension, which are bad here, then probably also like the properties that the strength that is involved in a dissection is also bad and poor, and so then the risk is higher. So there could be like a criterion of quality of the tissue that is really important. And well, whatever the stress in the tissue, I think that's we should be able to give to a surgeon a criterion. So this patient, what is the quality of the tissue in terms of mechanics? And then the surgeon could say, okay, well, I cannot leave this patient anymore with this aorta, because the aorta is from the day you're born to the day you die, it has to withstand with always the blood pressure and even the higher blood pressure for patients with aneurysm, with hypertension. But so if the tissue has become weak, then it needs to be changed. Okay, so for me, that's why still I think the tests we did were interesting, because they reveal some degradation of the tissue that is probably affecting all the tissue all around.
Yeah, fascinating. Really, really good. Thank you very much.
Thanks for your question.
Okay, so now we really need to move forward. Jordi, thank you very much for chairing this session. Stéphane, thank you again then for